Hey guys, so in this video we're going to discuss chapter 14. We're going to touch base on what the microbiome is, as well as get a little brief introduction into the immune system, and then we're going to go further into the immune system in chapter 15 and 16. So before we get started, let's start with the terrible joke. So the terrible joke is, what does a cell call a scientist? So the microbes, they call them paparazzi because they're always looking at them. So I thought that was kind of funny. But remember that. Okay, so that's going to be one of your bonus questions. Okay, so let's go ahead and jump into the chapter. So, the whole idea of this chapter is to go over the microbiome, explain what it is, also go into how it's diversified, and then also some, like I said, a little tidbits here and there of immunity, and then also the hygiene hypothesis. So with that, the human is pretty much, we're walking petri dishes. So just like how we know, the microbiome, it is all of the microbes that inhabitate the body. So it's not just like it's everything. So it's in the ears and the eyes and the gut in the on the skin, in the reproductive tract. It's everywhere. So that is what is considered the microbiome. And then also the reason why some of these are highlighted is because the highlighted species are the ones that are the predominant ones at the site, and also the fact that since they're predominant there, they have the potential of causing illness. And then also, as you see, there's a lot of staph that is highlighted. So that is, there are certain species of staph that they are differentiated by whether or not they can produce coagulase. And coagulase is an enzyme that helps coagulate serum, so helps it like clot. So generally, staph like aureus is considered coagulase positive, but there are some that are negative. And then there's also the species, the Staphylococcus, that is found on the skin. They're normally coagulase negative, and then so on. So this is kind of just to highlight some of the big ones that are in the area. So we're going to kind of touch base actually a little bit um, with all of these parts of the microbiome. We're going to touch base in them primarily in the chapters where we talk about the infections of that specific organ system. So with that, we're going to go into skin, because skin is pretty much our biggest organ that we have. Um, it covers your entire body. It is the first layer of protection, and it's extremely important. So there are a lot of features that come with the skin. And then primarily the skin is comprised of the epidermis. So if you take an anatomy, this should sound a little familiar to you. But with the epidermis, there are a lot of features of it that actually make it very difficult to be colonized on, meaning microbes, they have to have certain features in order for them to be able to grow. So the big thing is that the skin, it can dry. And a lot of times, bacteria need some kind of moisture. So that is why, if you think about it, you get more infections of the scalp, the ears, the reproductive tract, that skin area, because it does have some kind of moisture. And then also, the skin is actually acidic, not by much, but it is considered acidic. Most people are probably like around a pH of 5 to 6, but also depending on what skin area you're talking about can also to determine how severe the acidity will be. And then also with that, whenever we sweat, we produce a lot of salt. And with that, there is some microbes don't like high salinity concentration, so it makes it very difficult for them to grow. But most organisms that actually colonize our epidermis or the skin is gram positive. And that's because they tend to be more resistant and they can actually grow in the drier and the saltier climates of your body. So that's why you have a lot of staph epidermis on your skin. And then as well as Canada. So Canada can grow everywhere on your skin, but he does prefer to grow in the oral region of your mouth, primarily actually inside your mouth, but he can be around on the outside of the lips, and then as well as the vaginal area, primarily for females. But some men do have Canada in their general region. So definitely know the big highlights of the skin, like what characteristics do the microbes need to have. So generally they're gram positive, and then also they need to be resistant to salt and dryness. And the two big species are Staphylococcus epidermis and Candida species. So it's any Candida species. So it can be Candida albicans, be Candida aureus. Um, I want to say Fumigatus is one of them too, but I'm not too sure about that one. But definitely Candida albicans. He's the biggest one. He's the most common. Okay, so with that, we're going to transition into the actual anatomy of our skin. So our skin is comprised of three big chunks, essentially. So epidermis is the first layer, so that is what you can actually physically see. And then below that, we have the dermis. So the dermis is where like you have your hair fall, not your hair follicle, your hair like shaft and everything, and then also a little bit of blood vessels, not a lot, and then some fat, and then that's pretty much it. There's not a lot going on in the dermis, but it's the subcutaneous tissue that will house the like all the blood vessels, the sweat glands, and everything. So whenever like you actually get a cut or like a deep wound, and you actually, not like a paper cut, guys, like if you get like a paper cut, you're probably only going to the dermis. But what I mean is that if you like slice your finger or something with a knife, then you're going all the way to the subcutaneous tissue, and then you went all the way through your three layers of skin. Okay, so that's pretty much the big idea of skin. That's really all I wanted you to know. um yeah, that's pretty much it. Okay, so with that, we're going to transition into eyes. So the eyes, um, colonization of your eye is actually very, very hard. And that's because we have enzymes within our tears. So our eyes are very, um, if I'm sorry if you don't like the word, but moist. So our eyes are very wet if you think about it. Like you want them to be hydrated. And the reason why they're hydrated is because when we produce tears, and then as well as in those tears, we have something called lysozymes. And these are antimicrobial factors, meaning that they inhibit microbial growth. So, with that, it makes it very, very difficult for the eyes to be colonized. But it does happen. I mean, all of us have probably experienced pink eye one time or another in our lifetime, and that's completely fine. I mean, pink eye can be viral or it can be bacterial, and we'll actually talk more about that later on in the chapter, in a different chapter. But just like how our skin is colonized, there are um, certain organisms that can somewhat grow inside the eye, and the big ones are E. coli, Klebsiella, and Proteus species. These are all found in the conjunctiva, so that is a region of the eye. This is if you think of conjunctivitis, so that's like in your, if I remember correctly, my anatomy is a little rough, um, around the tear duct area. So that is whenever that's actually where you get an infection, is your in your conjunctiva. So um, that is the main area where you'll find microbes. And then also, like I said, occasionally viruses, they can cause pink eye, um, but normally it's normally bacterial, which is okay. It is treatable with antibiotics very easily. Okay, So definitely know, why is it hard to colonize the eyes? And it's hard to colonize the eyes because we have lysozymes within our tears. And then also know that generally speaking, it is colonized with Klebsiella or Proteus, and primarily in the conjunctiva of the eye. Okie dokie. Now, with that, we're going to go to another region. We're going to go to our oral and nasal cavities. So, with this is probably, there's a lot of microbes going on in this area because if you think about it, we're constantly breathing and ingesting stuff, and we're always being exposed. But within several hours of being born, so like if you, um, primarily if you're born vaginally, not so much cesarean, but through cesarean you can get a little bit of microbiome from the mom, but primarily through vaginal birth, because the baby is passing through the vaginal canal, and then with that, the baby is picking up all the microbes that were on its mom. And a lot of times that is how babies first get colonized. And then also babies can get colonized within their mouth if the mother like breastfeeds. So the baby has to latch onto the breast, and then with that, the baby can get colonized, probably primarily with Candida and some staph and some strep. But for the most part, the baby will be fine because it's actually good for the baby to be exposed to a germ or two. So um, that's primarily how the baby will first get colonized in the oral and the nasal region, is through birth, so through a vaginal delivery, as well as through breastfeeding after delivery, and then also continued breastfeeding as well. So what happens is that also as one develops, so whenever your teeth are maturing, with that there's also uh, bacteria, so primarily anaerobes, that are supported, meaning that as your teeth grow, these bacteria, they're also able to grow with your teeth, and they also start colonizing your these cavities. And then also there's some streptococcus species. These guys, they will adhere strictly to your oral surfaces, so like your tongue, your cheek, your gums. That's where they will adhere to initially, and then what will happen is that they will stick to each other. So they'll stick to each other, and then they'll form a colony within these cavities, which is perfectly fine because they're actually good for you whenever they're in check. And then also with the, so that was mainly the oral region. Now with the nasal region, the nasal and the throat region, you have the nasal pharynx, and then you have the oropharynx. So the nasal pharynx, this is the area that leads from the nose to the oral cavity. So that is um like back of your throat, I guess. I don't know how to describe it. So I don't know if anybody has ever had a nasal swab. It's super uncomfortable. Or if you've ever done like the neti pot, whenever like your sinuses are congested. So what you do like with the neti pot is that you literally just run warm saline through your nose and out the other nostril, and then that is that nasal oral region. That's where the saline is flushing through. I don't know if anybody has done that, but that is where it is. So it's the upper region, and then so this is the upper region compared to the oropharynx. So this is specifically the soft palate and the top of your epiglottis. So this is the back of your throat, the very, very top of the back of your throat. Um, and then these are specifically colonized by the following. There's a lot of them. I'm not going to expect you to know all of them, but definitely know maybe like max of three. But just know that the primary ones that are going to colonize these cavities is staph species, strep species. Those are the two primary, along with some anaerobic ones, but I'm not going to expect you to know all of these guys. So that is the oral and nasal cavity. So now we're going to go further down in the body. We're going to go to the um, to the to the heart region. But before we get to the heart region, we're actually going to talk about how dental is linked to heart health. So dental procedures, these can specifically cause organisms to go into the bloodstream. So, whenever like they are scraping off the plaque on your teeth with those little instruments, they create little micro abrasions, and sometimes the bacteria or the plaque can actually enter the bloodstream, and then from that can actually cause an infection called bacteremia. And a lot of times from that, it enters the bloodstream, and one of the first places it goes is your heart. And once it gets to your heart, a lot of times what will happen is that vegetation will occur. So the bacteria will adhere to the muscle or the tissue of the heart, primarily around like the valve region, and it will vegetate and grow and everything, and then causing um, endocarditis, which is infection of the heart. And it's very, very slow. It's very slow onset, meaning it doesn't show symptoms right away, which can be very, very scary because it's very difficult to treat your heart. If you think about it, it's very difficult. The two organs that are the hardest to treat is your heart and your brain. So a lot of times, once vegetation has occurred, it's extremely hard to treat your patient with antibiotics. So a lot of times, excuse me, it can be detrimental to your patient. So that's why they always say have good dental health, and you can limit the the likelihood of you getting bacteremia. So that's just a little sidestep for the heart for endocarditis. So with that, we're going to go into the respiratory system. So the respiratory system, um, there's some controversy on whether or not if the lungs are actually populated um, with bacteria, but in my opinion, there is some kind of microbes. Now, whether or not there's a large presence, I don't know that, but I definitely feel that there is some kind of microbes present, and a lot of times they will get cleared out. But just because they get cleared out doesn't mean that they didn't do a job prior to being cleared out. But the general belief is that the lungs and the trachea, so if you put your hand on your neck, you can feel that roughness. That's the beginning of your trachea, and then it travels all the way down behind your sternum, and then it branches off into your two lungs. So, generally speaking, there's like no harbor of microbiota in your trachea and your lungs because of the fact that that region, that organ system, is highly ciliated. So that is these guys. So these guys, they look like, to me, they look like those wacky inflatable arm men. So they literally are just like flapping around in your lungs to increase surface area so they can catch all the nastiness and make sure that it doesn't travel any further, and they clear it out. So that is why it's believed that the lungs and the trachea don't harbor um, bacteria because of the fact that it is highly ciliated and all the bacteria is being captured and cleared out. So that's why there's kind of a controversy, but I believe there is something that's going on there. But do I actually know what's going on? No, I haven't done the research. But to me, it makes it's funny to just have one organ system that is quote unquote sterile or clean, as the rest of your body has microbes. To me, that's very, very odd. Um, but just know the idea of why is it considered there's no microbes there, and then also what is the mucosal lining, what's the key factor of it, like what's the characteristics, and that's that they're ciliated. So ciliated means that they're just like little hairs, like little tentacles. So like I said, like those inflatable arm men that you see randomly on the side of the road, that's literally what they look like, constantly just trying to catch everything that enters. Okay, so that is the reproductive system. So now we're going to go further south in our body, and we're going to go to the stomach. So the stomach and the gut are the most diverse of all, in my opinion, um, aside from the skin. But the stomach, it is mucosal lining as well. It is much uh, less acidic and can harbor normal microbiota. But there are some that are extremely resistant to acidic environment, and that is primarily H. pylori. So um, he will not grow at that pH, meaning that um, he has to have an acidic environment in order for him to grow and proliferate and everything. And that's also how you get peptic ulcers. And then a lot of times, um, whenever H. pylori is out of control, like I said, it can lead to ulcers, and then sometimes that can actually even cause cancer. Um, Now, the actual cancer mechanism, I'm not too sure of. I need to look more into that, but just know that there has been documentations of if you have excessive H. pylori, then you are in a way predisposed to cancer. Okie dokie. So, with that, we're going to go even further. We're going to go to the intestinal tract. So, the intestinal tract that involves your small intestines as well as your large intestines. So, we traveled from the stomach and now we're in the small intestines, and then we'll travel to the uh, what's it called? The large intestines, and then like the colon and anus. So, the intestine tracts, we have something that is called pancreas secretions. So, this is primarily bile. And bile, what it does is that it will raise the pH level to slightly basic. And with that, we have decreased microbial content. So, with the fact that it's more basic, microbes, they cannot grow as readily. Some can, but some can't. And then primarily in the ileum and in the colon. So in the ileum and the colon, it is more acidic. So it's not very acidic. It's not like where it's pH of 4 or 3. It's around a 6-ish. So within the ileum and the colon, it is more acidic. So with the fact that it's more acidic, meaning that it has less bile salts, it is able to have a very, very diverse community of microorganisms. Okay, so remember that distinction. So that just that slight change in pH can definitely change um, your ecosystem of microbes. So in the intestines, almost all of them are anaerobic, and the reason why is because it's inside. It's inside your belly. Inside your belly, you don't have a lot of oxygen present, so they are going to be um, almost all anaerobic. So definitely keep that in mind. And then also the gut, the gut system can also include the liver and the gallbladder. It's not depicted here, and we're not going to really talk too much about the liver and the gallbladder, but just remember that for the gastrointestinal system, the liver and the gallbladder are included. But for the context of us, we're going to focus primarily on the stomach and the intestine region. And then also, why is our gut flora so important? So it performs numerous tasks. It does, as you can see, I think I only have 6 listed, but there are probably many, many more that weren't included. But the biggest things that they do, in my opinion, are definitely the immune system. To me, is the big one. We're going to talk more about the immune system later on in this chapter, as well as we're going to do 2 whole chapters of the immune system. And then also, what it can do is that it can ferment unused energy substrates, meaning it can metabolize them for us. It can aid in metabolism. And then also, the fact that we have good bacteria, with the fact that we have good bacteria, it can directly compete and prevent growth of bad bacteria in our gut. And then with that, You can The good bacteria can help in regulating the development of our gut, so it can constantly keep our gut developing and going in the right direction. And then also it can aid us in making vitamins that we actually can't. So it will make the vitamins for us, and then we'll end up, from the vitamins that they make, we'll end up utilizing them. And then also producing hormones to direct the host in order to store fats. So, with the fact that these microbes are producing hormones, once we sense that they made hormones, then we can actually, our body will recognize it and be like, oh, we need to do something. So, it's a good signaling uh, mechanism. So, the bacteria can also help guide us in making our body healthy. So, it's huge, huge, huge. Okay, so we've gone through the colon, so we've gone all the way from the skin, we've gone from the mouth to the lungs, to the stomach, to the intestines, and now we're going to go to the general to the genito urinary tract area. So where we urinate, and then also reproductive. So the urinary bladder, its main function is to hold urine, um, to temporarily store it, and then we can secrete it. So it was originally thought that pee was actually sterile, but it isn't. Pee is not sterile at all. Um, it does harbor some anaerobes, because if you think about it, your bladder, all of our organs inside, for the most part, the organisms inside of our body that grow inside of our body are going to be anaerobic. So same thing with the bacteria that grow in the urinary bladder. They are going to be primarily anaerobic. In the urinary tract, there is something called the distal urethra. So this is where uh, the urethra is where your pee will travel out of your body and everything, and um, that is where the distal urethra is part of where it's right before the pee will actually exit your body. So for men, the distal urethra is primarily in the beginning of the penis shaft and then a little bit upward. For females, our urethra is definitely different anatomically speaking. We'll go more into anatomical differences whenever we go into actual infections of the genital region. But with for females, it's all internal for us, whereas some of the male urethra is, I don't want to say external because it's not external, but it's through the penis, whereas women, it is actually part of in our lower abdomen. So the distal urethra, so that is where it is, part of the urethra that is closest to the outside of our body, that is generally colonized with staph epidermis primarily. And then also with the fact that it is slightly colonized, it can actually cause UTIs. And then we'll talk more about UTIs later on. But if a UTI, if it progresses into a bladder infection, it can cause cystitis, which is a very, very, very painful infection. So this is where the infection traveled up your urethra into your bladder, and then sometimes it can travel even further up into your kidneys. So, but like I said, we'll go more into that detail later. But just know that um, this region is primarily colonized by staph epidermis and then enterococcus species. Those are the two big ones. So that's with the urinary. Now with vaginal secretion, so men so much don't really get a lot of infection of their reproductive tract solely because of their anatomy. Um, but with women, vaginal secretions, they are acidic. So with that, with the fact that our vaginal region is acidic, a lot of times growth of microbes doesn't occur. But there is one specific one, bacteria wise, that will grow, and that's Lactobacillus acidophilus. So he will grow in our vaginal tract area, and that is because he's acid tolerant. And then also Canada species can grow as well. And then with lactobacilli in Canada, they're harboring the same area. They keep each other in check, and then our our machinery is happy. So we have a happy reproductive system. So there's a balance between primarily lactobacilli and Canada species. There are also other species, but those are the two big ones. There's a balance between them, and with them being kept in check, it prevents infection. Now, um, if let's say that you had a slight um, you had a bacterial infection and you start taking antibiotics, you take a broad spectrum antibiotic. Well, that broad spectrum antibiotic is going to kill all your bacteria. That's including your gut flora and your vaginal flora. So, with that, a lot of times some women, not all, will experience a yeast infection while taking antibiotics. And that's because the lactobacilli is being killed off. So, with no lactobacilli, Canada can like grow out of check, and then Canada will cause a yeast infection. But we'll go more into detail about that later on. But here is the anatomy that we've been talking about. So this is a female. So we have our vagina region. So this is where like the lactobacilli and the Candida will reside. And then our urethra is very, very short, whereas males, they have a longer urethra. So that is partly why women are more prone to UTIs as compared to males. And that's why they always say make sure you urinate after intercourse, just to help flush out any bacteria that may have happened from sexual activity. So, but like I said, we'll go more into detail about that. Um, so this is what I was talking about. So in up here it said that the distal urethra, so that is the proximal, so that's close to outside. But then UTIs, what can happen is that they can travel their way into the bladder. So with that, what happens is that you start with having just a urinary tract infection. So it's primarily an infection of your urethra. That's where mainly it happens. But sometimes what can happen is that bacteria can travel up into your bladder, cause a bladder infection, and then if it's not treated properly, then that bladder infection can ascend even more, can ascend up your ureters into your kidney and causing a kidney infection. So it's extremely painful. But this is a general anatomy that we've been talking about. The only difference is that males they have a longer urethra as compared to females. That's the main difference. Okie dokie. So now we're going to talk about why is our microbiota so fabulous. So our microbiome is extremely fabulous. It's very, very beneficial for us. We do have a mutualistic relationship with our bacteria on our body and inside of our body. So what do? They can help us digest food. They can help us synthesize vitamins, and they can also interfere with other pathogens colonizing us. And through that is through three different mechanisms. So they can actually actively be competing for attachment sites. So like attachment sites on the skin or in the stomach or in the vaginal region. So if the good bacteria is already adhered, the bad bacteria can't adhere. Or they can actually be directly competing for food. So if the good bacteria eat all the food before the bad ones, then the bad bacteria can no longer survive. And then also, they can actually be synthesizing antimicrobial compounds. So the good bacteria can be making something that the bad bacteria doesn't like. And with the bad bacteria not liking what they're making, then they cannot thrive. And then with that, it helps shape our immune system, and this is primarily through producing something that are called immunodullins. So these immunomodulants, these specifically, they'll modify secretions of cytokines. We'll go into detail what cytokines are, but the big picture of cytokines is that they are used for a lot of things. They are you can think of them as a signaling factor, and they help regulate. They send messages as to what should a cell do. But we'll go more into cytokines a little bit later. I'm not worried about the last bullet point. So with that, let's go ahead and move on. So that's the benefits of it. Now, what are the downfalls of having a microbiome? So, what can actually happen is something called accidental penetration. So, this is where a microbiota is normally in one region, but for somehow, some way, it got outside of its environment, and with that, it caused a serious infection. Possible reasons for this penetration is something called opportunistic pathogens. So, opportunistic pathogens are exactly what they sound like. They are normally not pathogenic to us, but whenever they are not kept in check, they will cause infection. So, that can be like if you had a surgery, or if you are on like immunosuppressants, or if you're somehow immunocompromised, like if you're undergoing cancer treatment, radiation, if you're HIV positive, that can also make your immune system weakened. And with that, it predisposes you to pathogens that are normally not pathogenic to the normal population. So that's the main risk of having a microbiota. So with that, how do we keep our microbiota from actually going into different regions? So we do that through three different ways. We do that through epithelial barriers, the blood-brain barrier, and the placental barrier. So the placental barrier is only for females, and this will only happen whenever the female is pregnant. Okay, but the blood-brain barrier and the epithelial barriers, that is present in both male and female. With the epithelial barriers, this is specific. So I'm actually just going to go to the next slide for that. So the epithelial barriers, it is a layer of epithelium, so a layer of skin cells. They're held together by something that are called tight junctions, which is this region right here. So these are all individual cells, and they're held together by these tight junctions. And with that, it's kind of like a glue for the cells, so they can attach to each other and attach to the cytoskeleton of the cell. And with that, tight junctions, they're regulated, and they will only open and close whenever they are given a signal. So think of them as border patrol for epithelium, for the skin. So they will only allow certain things to go in and out with permission. So they have to have the right papers in order for them to travel from cell to cell. So that's the whole purpose of the epithelium. Now with the blood-brain, this is specifically for the blood-brain barrier. So if you think about it, you don't want anything just getting to your brain. Like your brain is the computer. You don't want anything getting to the motherboard. So this one is very, very, very selective. It is highly selective in the fact that it only allows the essentials to pass through without with minimal regulation, and that's primarily oxygen and sugar. Those are the two main things. Everything else, like if it's larger than those two things, it will be blocked. So everything from hormones to neurotransmitters, and then including viruses and bacteria, a lot of times they're refused access into the blood-brain tissue because of the fact that they're not able to travel because they don't have the right papers. Now, there are some instances where they can pass the blood-brain barrier, and we'll talk about that whenever we get into the actual infections. But just know, for the most part, they cannot. It's very, very difficult for them to go into the blood-brain barrier, but it is possible. It is possible. So with that, that is why it's very, very hard to treat bacterial infections of the brain, as well as fungal infections, parasites, everything. That is because antibiotics cannot pass the blood-brain barrier. They are too big and too bulky. So that is why it's extremely difficult to treat bacterial infections of the brain. So this is just a demonstration of how tightly regulated it is. So this is just a pretty picture. I'm not going to expect you to know the anatomy because we're not in an anatomy class. But just know that tight junctions are also present in the blood-brain barrier. Okay? So, one last thing on that before we go into placental. So there are, like I said, there's a handful of things that can definitely flow in and out of the cell very, very easily. And that's primarily oxygen, carbon dioxide, and actually alcohol. So that is why you get intoxicated very, very easily. That's because alcohol can actually travel into your brain very freely. Now, other things that are bigger than these molecules can pass the blood brain barrier, but they have to go through border patrol. So that's primarily glucose, hormones, primarily L-DOPA. So L-DOPA is the precursor to dopamine, and dopamine can't cross the blood-brain barrier. So you have to have a precursor in order to make dopamine, and that's the same thing with tricin. Same exact thing. This is a precursor for serotonin, and serotonin can't cross either. So, what you want though, you want the L-DOPA and you want the tyrosine to be able to travel into the brain. But then once you make serotonin and once you make dopamine, you don't want that stuff traveling outside of the brain region. Okay? So, with that, we're going to go into our last one, which is the placental one. So, this is the placenta. This is where the umbilical cord is, and then here's the fetus. So, this is just an overview of what it is. So, this image shows the overview of the maternal and the fetal circulation. So, as you see, fetal and maternal blood, they do not mix. So what happens is that the maternal blood will enter this intervillous space that is surrounded by the villi, which contains these fetal capillaries. And then what will happen is that the fetal capillaries, they will merge all together, picking up all the maternal blood and everything, and then it will go into the umbilical cord, into the umbilical vein and artery, which will then transport all the necessary nutrients to the fetus. So it doesn't go directly from mother to baby, it has to go through border patrol again. So it has to go through this intervillous space. So with that, that is this area right here, kind of zoomed in. So with this is showing the placental membrane and the fetal capillaries. So the fetal capillaries are the red, the placental membrane is all the way around. So this is why, specifically why maternal and fetal blood don't mix, is because of that membrane. So you don't want mixing, you want to minimize what can get to the baby, because if anything and everything can pass between mother to baby, then there will be a lot of diseases that are happening. Or what if the mother gets sick, gets a bacterial infection, then the bacteria can go to the fetus. So that is why we have these measures. Okay, so with that, now we're going to transition into the immune system. So this is just going to be a little dribble of our immune system for chapter 15. So we're going to talk about the innate immunity. So the innate immunity is includes the mucosa, the brain fart, the epithelial cells, and then the tight junctions. So this is where it keeps all of your normal microbiota in place. And then what happens is that whenever breaches occur, so whenever something goes somewhere where it's not supposed to be, our immune system is going to react. And there are two different ways that our immune system reacts. It will react either in something that's called non-specific, which is the innate immunity, or adaptive. So innate immunity comes first, and then we can get adaptive immunity. But innate immunity is the process where the host will recognize the presence of something that's not supposed to be there and produce a generalized response. And then from that, we'll get something that is called adaptive immunity. This takes a little bit longer to develop because it's more memory immunity. And this is where we're actually producing antibodies and then also specialized immune cells. So if we are ever exposed to that pathogen again, we can have a better response and a more specific response. But we'll go more into detail about that in the 2 chapters to follow. Okay, so with that, I do want to touch base on something. So, um, this image might look very scary, but I'm gonna break it down. So this is just our cell. Okay, this is a very simplified version of our cell, and we have these receptors on our cellular membrane. And we're going to talk specifically, these are called TLRs, or toll-like receptors. So these are receptors. So these are on our cellular membrane, and they will recognize something from a pathogen. So like TLR2, it recognizes more than this, but it mainly recognizes lipopeptides, whereas TLR3, it will recognize double-stranded RNA. So if it recognizes double-stranded RNA, it can recognize some viruses. TLR number 4, it recognizes lipopolysaccharides. And the fact that it recognizes lipopolysaccharides, that means it's more specific for gram-negative bacteria. Okay? Because gram-negative bacteria, they have lipopolysaccharides present on their cellular membrane. Okay? TLR5 will recognize flagellin. TLR9 recognizes microbial DNA. Now, whenever any of these TLRs are bound, so let's say that we have a gram-negative infection. So let's say, like I said, I have a gram-negative infection. They have lipopolysaccharides on their outside, and then that will bind to the TLR4 specifically. So as you see, like this is a triangle, like a point. So this lipopolysaccharide is specific to TLR4. Whereas if lipopolysaccharide trying to bind to TLR3, it would not happen because they are not the same shape. So it's like trying to put a round peg in a square hole. It won't happen. So it has to be perfect. You have to go with round peg, round hole. Okay? So let's say that I have binding. Something's going to happen. A signaling cascade is going to happen. And that signaling cascade is going to travel to the nucleus. The nucleus is then going to be like, oh my goodness, I'm under attack. And then from that, it's going to transcribe and translate cytokines. And then these cytokines are going to be released. And then these cytokines, what they do is that they send out an immune response. They are immune signals, and they're just like, help, help, we are under attack. Essentially, that's the very, very, very general overview. But just know that cytokines are released, as well as other immune proteins. But for the most part, just know about cytokines right now. And what will happen is that will stimulate inflammation, or it will inhibit inflammation, whatever it may be. So there's a lot of things that cytokines can trigger eventually. But we won't go into detail about that because that can be a whole immunology course. But just know that cytokines are they will most likely either promote or inhibit inflammation. Let's just go with that. Okay, so that's just a little side step. We'll go more into detail about that later, but that was just a little side step. Okay, so now we're going to go into the concept of how do these microbes talk. So we sort of already talked about this a little bit, but we're going to touch base on it again. So the idea is that if microbiota talk, can they talk back? So generally speaking, yes. So there are a lot of chemicals that are being released, and all these chemical changes, chemical regulations, productions, inhibitions, it is considered to be a conversation. And then this is where, in that conversation, genes are being regulated. And sometimes it can be for a mutual benefit, assuming that it's our microbiota that is talking and we're talking back. Or it can actually be a bad benefit where it's the bad microbiota, like the bad pathogens that are hurting us. But specifically, like in mice, there was end products that were made from microbial metabolism. So, like how we talked about microbes, they can help support making vitamins. So that can be considered an end product. And a lot of times that was found in blood, and then the fact that it was thought that blood was sterile and so was urine, it kind of uh, opened up this door of like, oh my goodness, like we need to change something. It's opening our eyes into something all new. So that is what um, why there's a lot of researching going on of like how do we talk, how does the host and the microbes, how do they talk, and with that it changes um, the whole picture. And then with that, there's a lot of changes in microbiota makeup. So the microbiota is not stagnant, meaning that it will respond to like whenever you're stressed or whenever you're going through puberty. So you had a different microbiota as compared to whenever you were 6, as compared to when you were 16, and then compared to when you're 25, and so on and so forth. Your microbiota is always changing. So, whether it be due to stress or hormones or environment, diet, it's always changing. So, just keep that in mind. Okay, so I'm gonna be honest, I don't even know why I have this even on here, so we're gonna skip it. So, how do they actually communicate? So, we've talked about this through quorum sensing. We've kind of dabbled in this a little bit, but with quorum sensing, I just want you to know that quorum sensing has to do with the ability to detect, and then after detection is there, it's followed by a response, and it's specifically followed by a response to cell population density, and from that, genes can be regulated. So, for example, um, quorum sensing will enable bacteria to restrict expression of certain genes based on if there's higher density of the population. And with that, it can result in phenotypes that can be beneficial for them. So just know that there is always crosstalk between them. So it's not always just going to be E. coli to E. coli. It can be E. coli to staph to Candida to lactobacillus. Just know that they're all talking just like how we do, but just in a different language. But definitely know quorum sensing, okay? Okie dokie. We are almost done, guys. So, like 3 more slides, I think. Okay, so why is the gut so important? So, the gut is extremely, extremely important. I can't stress it enough. Um, so, this is just an image, like a very generalized overview as to like the gut is the biggest organ of the immunity system, of the immune system. And actually, there's a video. I will show this in class, but it's also in the PowerPoint for your knowledge. I'm not going to show it just for time's sake, but definitely look at this. It will go into detail as to um, why is the gut very important, um, why is the microbiota so diverse, and then also primarily with the immunity, gut immunity. That's the big picture that I want you to get from that video. Okay, so one more slide. So with that, I want us to just take a little sidestep into probiotics. So there are a lot of application of probiotics. So the whole idea of probiotics is to restore balance and to diversify the microbiota environment. So that's the whole purpose of probiotics. So probiotics are the good guys. These are the ones that you want a lot of. A lot of times, like that's why doctors will say like whenever you're taking biotics, make sure you're taking probiotics and eating yogurt and everything in order to help your gut not be so raw, because you are killing your gut bacteria whenever you are taking antibiotics. So the whole idea of taking probiotics is to help regulate your digestive tract and then also to restore balance. And then a lot of times, if you give kids probiotics whenever they have like diarrhea, it can also help ease the symptoms and then also um, shorten the duration of diarrhea. Now, the effects if your microbiota is disappearing. So that brings us to the hygiene hypothesis. So this is as human health has um, improvised or improved, so improved, diversified over our years and throughout different countries, there have been unexplained new diseases that have arisen. So there's one hypothesis. So this is the idea. It's called the disappearing microbiome hypothesis, or known as the hygiene hypothesis. So this is the idea of that since we are now becoming more conscientious of our surroundings, we're starting to become more hygienic, which is good. But in that, the fact that we are being overly hygienic or scared of all these bugs, we are actually decreasing the diversity of our microbes. So, such as like a lot of times parents are like, oh my God, don't play in the dirt because they're afraid of the kids getting sick. But whereas now the fact that the kids aren't being exposed to the microbes in the dirt, now the kid is going to not be as diverse. And with that, their microbiome is not going to be as robust. So that's kind of just the idea. That's just a little side step. I'm not 100% concerned about it, but I do just want you to know the big picture of it. So with that, we are done. That was a very, very long lecture, but I'm glad y'all stuck through it. So we talked about the normal microbiota throughout various regions. We also talked about the benefits and the risk. We talked about the microbiome barriers, as well as how do they talk, excuse me, and then also the theory of and the application of probiotics. So with that, I'm going to leave us on a funny picture. So, who else loves our immune system? Antibody? I thought that was kind of funny. Anyway, that's a little nerdy stuff. So, that will be another bonus question, guys. So, what kind of animal is this? It's an eel. Woohoo! Okay, so with that, guys, I'm gonna leave you to it. I hope y'all enjoyed this lecture. If y'all have any questions, don't hesitate to contact, and y'all have a good night.